Hi, thanks for watching my talk. My name is Noori Choi at the University of Nebraska-Lincoln. I will present our research about the effects of signal complexity on the copulation success of Schizocosa stridulans wolf spiders. The complexity of animal communication that are using multiple signal components within and across sensory modalities has long been studied by researchers. And one of the fundamental questions is, why do animals use complex signals? Traditionally, biologists have tried to answer the questions by ablating or adding signal components through the manipulation of signaling apparatus and observe whether manipulation induced any variation in the receiver's behavioral or neural responses. In this approach, signal complexity has been regarded as the size of the repertoires or the number of discrete components. However, this traditional approach only can assess the functional variation among discrete components, but cannot investigate the emerging properties by the complexity of the interactions or temporal orders among signal components within a sequence of signals. Thus, recent studies have started to introduce the complexity metrics from computer science, such as Lempel-Ziv complexity, Shannon entropy, or entropy rate, to investigate the effects of the signal complexity on animal communication. This approach can test the functions of various aspects of complex signals, including the number of components, temporal orders, or interactions among components. However, this approach often requires labor-intensive and time-consuming works to classify and annotate unit signal components to convert animal signals into a sequence of discrete labels, to compute the complexity metrics, like binary codes in computer science. Given the difficulty to get enough sample size through the labor-intensive jobs, many researchers have tried to automate the process using machine learning algorithms. However, the recent progress has been predominantly focused on some vocal animals which have well-documented signal repertoires for the verification of approach. In the present study, we aim to broaden up the scope of the interdisciplinary approach to another well-studied but non-vocal communication system: substrate-borne vibratory signals of Schizocosa wolf spiders. Schizocosa wolf spiders have several benefits for this project. First, males produce the substrate-borne vibration as a major channel for their sexual communication. And in some species, the complexity of vibratory signals influence the copulation success of signaling males. Also, with the long history of study, well-documented signal repertoires are available for the verification of the automated process we developed in the project. These videos are the courtship signals of two Schizocosa wolf spider species. Our study species, Schizocosa stridulans, males produce the multimodal courtship signals like many other species in the genus. Schizocosa stridulans males produce the three vibratory signal components: revs, idles, and lag taps. Rev is produced by distillation using pedipalps and tremulation by abdomen. And this is the rev sound. And idle is produced by percussion by four-leg taps and stridulation using pedipalps. And this is the idle sound. And leg tap actually is the first part of the idles, which is the percussion by four-leg taps, is only rarely occur during their courtship signal. And this is the leg tap sound. Also, idle and leg tap are associated with the visual signal components, four-leg tapping. And there is evidence that the visual courtship rate is critical for the mating success of this species. In this project, we have three major objectives. First, develop automated algorithms to identify and cluster vibratory signaling components. And the second, to test the effects of the sequence-based signal complexity on mate success of the Schizocosa stridulans. And the third, verify the application of the information theory through the comparability with traditional approach to measure the signal complexity of Schizocosa stridulans. To investigate the effects of the signal complexity on mate success of the Schizocosa stridulans, we first conducted a matrix experiment and recorded the male courtship signals during the experiment. The spiders were given 5 minutes to interact with each other, after 5 minutes of acclimation time for females. After the experiment, we recorded the mating success of the individual males. For the first objective, we developed a Python program to automate the whole process of the signal analysis from noise filtering to finding discrete clusters of the signal components without any given training dataset. We expect that the unsupervised clustering method will be useful for other animals using vibratory signals that have a little prior knowledge about their signal components. In this presentation, I'll present the last 3 steps only due to the limited time. If you have any questions about the first 3 steps, please leave a comment. In the many automated signal classification study for vocal communication, researchers usually use spectral characters such as dominant frequency as a major feature for further clustering or classification. However, due to the higher rate of the degradation or attenuation by substrate, vibratory signal components often do not show a consistent spectral characters, even within an identical signal component. As you can see in the spectrogram on the slide, even the first 3 RAP signal components of Schizocosa stridulans do not show a clear pattern in the spectral characters in the spectrogram. However, the pattern of the amplitude variation, blue RMS curve, is relatively well conserved than spectral characters. Thus, we decided to use the pattern of the RMS variation as a major feature for the clustering algorithm. To extract features of the amplitude variation, we calculated the dynamic time mapping distance of each unit signal components with 9 reference triangular membership functions that vary in the location of peaks. The triangular membership functions have the same length as each unit signal component. After calculating dynamic time mapping distance with reference functions, we normalized the dynamic time mapping distance by jet normalization to control the effects of the variation in the length among unit signal components. In this graph, you can see the different signal components by manual classification. Idle, lag tap, and RAV show a clear difference in the pairs of the dynamic time mapping distance with reference functions varying in the location of peaks. Idles were most similar to the reference functions that has the peak at the 10% of the signal length and increase with the peak location. But RAVs were most similar to the function that has the peak at the middle of the signal, and lag tap shows the overlap distribution due to the limited number of the unit signal components. Using the extracted features, we calculated cosine similarity among unit signal components, and then run hierarchical density-based spatial clustering of application with noise, aka HDB scan, to find a discrete signal component. HDB scan has several benefits for finding discrete components that are unbalanced in the proportion in the total data set. In the T-SNP plot, you can see the 2 clear clusters of RAVs in blue and idles in orange with several gray dots of noise. Also, the comparison metrics between the manual classification and HDB scan shows that the results of the unsupervised clustering produced a similar result with the manual classification of more than 7,000 unit signal components. Through the first objective, we show that the automated signal processing was able to find discrete signal components that are similar to those that were previously defined by experts. However, the automated process does not fully undermine the role of the experts in data analysis, given that the HDB scan failed to find a discrete cluster for rarely occur lag tap sum. For the second objective, we converted courtship signals of individual males into a sequence of the labels of the 3 different signal components. For accuracy, we used the manual classification results at this step. Using the converted sequence, we calculated 3 different metrics. Lempel-zip complexity, which is the number of the distinct patterns that exist in the sequence. and Shannon entropy, and entropy rate based on the first-order Markovian process. Due to the effects of the signal length on the Lempel-Zip complexity, we normalized the Lempel-Zip complexity using the formula from Zhang et al. 2009. Then, we conducted statistical tests to investigate whether the signal complexity has an impact on the mating success of the males. To verify the comparability of the metrics with the traditional approach, we also calculated the rate of the vibratory and visual signals of the males, which have been used as a proxy of the signal complexity in this spider. Through the binomial logistic model, we found that the vibratory signal complexity has a significant effect on the mating success of males, regardless of the metrics. Copulated males produce the more complex signals than non-copulated males. As the previous study suggested, we found a significant effect of visual courtship rate on the mating success. But general vibratory courtship rate did not have an effect on mating success. Thus, copulated males produced more visual associated vibratory signals, like idles and leg taps, than non-copulated males. Besides the major objectives, we found that the copulated males produced the more visual associated courtship signals when the encounter to females were heavier. But non-copulated males did not show such a pattern. Complexity metrics also shows a similar patterns, but the effects of the interactions between mate success and female body mass did not significant on those complexity metrics. So, in the presented study, we first developed the automated algorithms for finding clusters of the vibratory signals of Schizocosa off spiders using non-spectral characters. Also, we show that the complexity metrics from information theory produce the comparable results with the traditional approach about the effects of the signal complexity on the mating success. We expect that the application of the complexity metrics will allow researchers to compare the complexity among different species, which is difficult with the species-specific metrics such as visual courtship rate. Also, interestingly, we found evidence that the males may alter their signal complexity depending on the body conditions of encountered females. I want to thank my co-authors and Hebets lab members and UNL Behavior Group, and our funding sources. Here are references of this study and the Python packages used for the development of automated process. Thanks for watching. Please leave a comment if you have any questions.